In the quiet corners of human habitation, a stealthy intruder lies in wait, the Triatoma infestans, commonly known as the kissing bug. Join us on a revealing journey into the secretive world of this blood-sucking insect, as we delve into its evolutionary history, ecological significance, and the challenges it confronts. Lurking undetected within the cracks and crevices of substandard housing across parts of South and Central America is one of the world's most insidious insect vectors, the kissing bug. This innocuous-looking bloodsucker plays a devastating role spreading the deadly parasitic Chagas disease that afflicts millions. Yet despite its menacing behaviors and fearsome disease associations, the kissing bug represents a unique, highly specialized life form humanity largely brought upon itself through ecological disruptions. This documentary explores the fascinating biology, behaviors, and complicated existence of the infamous Triatoma infestans. Evolutionary history. Kissing bugs belong to the insect subfamily Triatomine, a group of around 150 species divided into 19 genera and 9 tribes under the Reduviidae family of assassin bugs. Genetic evidence indicates triatomines diverged from other reduviids over 100 million years ago in the late Cretaceous period alongside the diversification of mammals they eventually evolved to feed upon. Fossil evidence shows early triatomines were likely scavengers feeding on decaying organic matter before some lineages transitioned to exploiting vertebrate blood sources and forming endosymbiotic relationships with Trypanosoma parasites like those causing Chagas disease. This transition may have occurred in the Oligocene around 30 million years ago among groups ancestral to modern kissing bug genera. Triatoma infestans represents just one species within the highly speciose Triatoma genus containing over 70 known types. Triatoma infestans branched off relatively recently from a common ancestor shared with the Mechus group of closely related kissing bug genera. Genetic data suggests the Triatoma infestans lineage emerged around 8 million years ago in the Miocene as its ancestor began dispersing northwards from an Amazonian basin origin point. Morphological description. At just under an inch long, adult Triatoma infestans resemble many other sizable true bugs with their flattened bodies, conical heads, compound eyes and rostrum mouthparts adapted for piercing skin and drawing blood meals. Dark brown or blackish coloring allows easy camouflage within dim household environments. While incapable of flight, their bodies are well-adapted vectors. Highly developed chemoreceptors allow tracking expelled carbon dioxide to precisely locate warm-blooded hosts for feeding. Short legs allow quick mobility over surfaces. Enlarged rear legs enable leaping away from threats. Kissing bugs derive their name from a curious habit preferentially feeding around the eyes and lips of sleeping human victims where facial skin is thinnest. During nocturnal feeding sessions, they inject a local anesthetic numbing the bite while transferring feces containing infectious Trypanosoma cruzi parasites. These parasites are then able to enter the victim through the bite site to initiate chronic Chagas disease infections. Ecology. Kissing bugs play a natural ecological role as hemimetabolous predatory insects and blood meal scavengers in their native South American environments. Their ability to vector Chagas pathogens to humans emerged only after colonial-era rural housing allowed population expansions in close human proximity. Despite their infamy, Triatoma infestans and other kissing bug species help control populations of insect pests, decaying organic matter, and small invertebrates in regions they originally occupied. Transferring Trypanosoma cruzi between native mammalian hosts enables lateral transmission and genetic exchange for the parasite. However, their evolutionary transition into domestic habitats radically transformed their ecological impact. Access to food supplies of human blood, livestock, and lack of predators allows exponential population growth and disease spread. Global dispersal thanks to human trade and travel then exported local epidemics on a pandemic scale. Habitat and behavior. Once confined to sylvan settings and animal nests, Triatoma infestans now exhibits a strong proclivity for sheltered man-made structures and human settlements across its endemic range. Dirt floor housing materials allow easy access into cracks and roofing spaces immediately adjacent to concentrated blood sources of local residents. Scavenging insects like cockroaches even facilitate transmission by consuming kissing bug feces laced with infectious Trypanosoma cruzi parasites and transferring them to other surfaces. With no cleaning regimens and poor sanitation awareness, domiciliary cycles of Chagas infection sustain themselves indefinitely within communities. At night, kissing bugs emerge in response to warmth and carbon dioxide cues signaling when residents are asleep and easy prey. Their specialized mouthparts allow injecting a numbing agent, enabling stealthy consumption of blood meals over the course of 20 to 30 minutes undisturbed. After feeding, kissing bugs defecate, often unwittingly passing Trypanosoma cruzi parasites into their own fecal spots or the bite wound itself, resulting in ensuing infections. Chagas then enters the chronic asymptomatic phase, lasting decades before potentially fatal heart or intestinal complications emerge unless treated. Reproduction occurs exclusively through egg cases called pillars, containing around 40 eggs each, carefully deposited in secluded household refuges for protection. After emerging, nymphal instars must shed their exoskeleton up to 5 times while acquiring enough blood meals to eventually metamorphose into fully winged adults. Conclusion. In many respects, the kissing bug represents an archetype for ecological imbalances where human behaviors actively enable zoonotic disease vectors to thrive in domestic environments. Regional Chagas epidemics remain widespread due to political instability, poverty, inadequate public health infrastructure, and most critically, a lack of sustainable housing and living conditions that would deny these insects disease reservoirs. National initiatives supported by the World Health Organization and partners in endemic countries aim to eliminate Chagas through insecticide spraying and community outreach. Yet the kissing bug's resiliency always allows repopulation unless structural housing conditions improve to fully block their domiciliary life cycle. Despite their fearsome status, the evolutionary backstory for how kissing bugs became infamous disease vectors also offers lessons around respecting natural ecological boundaries. Only by fostering stable communities and sanitary housing can we disrupt the domestic transmission cycles that let insects like Triatoma infestans proliferate so destructively as an inadvertent consequence of our own behaviors.